protein becomes the actual ribosome. And so with those four things, you can then actually turn your RNA into a protein. And those processes of transcription and translation are basically the same in all cell types, with four important differences. So what are the differences in those processes between bacteria and eukaryotes? Bacteria do not have a nucleus, and why is that important?
They can start
No delay between transcription and translation. What else?
They don't have operons.
They don't have, the bacteria use operons. And operons are multiple genes, one promoter. What else? Bacteria use polysomes, which are multiple ribosomes. And lastly, bacteria do not have introns. What are introns? The internal non-coding sequence. And so, what is the overall effect of those four differences between bacteria and eukaryotes?
much faster growth.
Bacteria make their proteins faster, allowing them to grow faster. Good. What else would a cell need?
FTSE.
FTSE, what is that used for? Division, what is the other major cytoskeletal protein that they have?
MREB.
MREB, what is that used for?
Growth and elongation.
Growth and elongation. I also heard someone say cell wall. So what is the cell wall? What is it used for? It's a protective layer around the cell. And there are two types of cell walls that are commonly found in bacteria. And what are those two types?
Gram-positive, gram-negative.
Gram-positive, gram-negative. So if you were to describe the structure of a gram-positive cell wall, what does it contain?
Thick peptidoglycan.
Thick peptidoglycan layer,
Yeah.
TA and LTA, and then
Proteins.
some proteins. But it's really that peptidoglycan molecule that is the defining characteristic of a gram-positive cell wall. And so, how do you make peptidoglycan? Well, that's going to be lipid A. Get back to that. How do you make peptidoglycan?
Nagging them with a beta 1,4.
Alternating NAG and NAM connected by beta 1,4 sugars.
The amino acids.
And then the four amino acids that come off of them, which are L-ala, D-glutamine, M-dap or L-lyse, and D-ala. So what do the L versus D mean in the amino acids?
Direction.
We have the direction, so we have the left and right-handed. And then what is the M-dap or L-lyse significance? So that's the direct or indirect cross. M-dap allows for a direct cross bridge. Lysine requires an indirect cross bridge. So that's the gram-positive side. How about a gram-negative cell wall? If you were to describe that in words, what does that contain? Thin peptidoglycan layer, and then an outer membrane with LPS. And then there's some pores and proteins and stuff. But really it's that LPS molecule that is the defining molecule. And so, what are the parts of LPS?
O-antigen.
The O antigen on the outside, and what is that? Species-specific sugar. We have the core polysaccharide inside of that, and that is 2 KDOs, some other sugars. And then inside of that, the lipid A molecule. And what is lipid A made of? So you have 2 sugars with phosphates, and then at least 5 fatty acids. Now, lipid A can be an endotoxin. What does the term endotoxin mean? So it's normally inside the cell. If it gets broken down, it can then be released and cause septic shock. But the O antigen also helps us prevent infection because it activates an immune response. Good. What else does our cell need?
Flagella.
Flagella. What is the function of a flagella?
Motility.
And that's true in all cell types, but the structure of a bacterial flagella is very unique. So what are the 3 parts of a bacterial flagella?
Basal body.
Basal body, hook, and filament. Now the filament is the tail part that actually generates all the movement, but all of the control happens down at the basal body level. So what are the 3 functions of the basal body? Anchor, movement or motor, and switch. What protein serves as the switch?
Flea.
The flea protein. And what does flea switch between? Clockwise and counterclockwise movement. And that results in what process? Random walk. So describe the process of random walk.
Run in a straight line. Tumble.
So flea alternates between clockwise and counterclockwise movement, which results in alternating between runs and tumbles. Runs are movement in a straight line. Tumbles are changes in direction. What happens if the bacteria wants to go somewhere? Like let's say there's a chemical attractant in the environment and it wants to get there. Taxis, right, or chemotaxis in that case. And so taxis is a biased random. Good. What else does our cell need? PMF. What is the PMF?
Electrochemical gradient.
Proton motive force, or the electrochemical gradient, and it's used to
Power the cell.
power the cell, makes energy for the cell. How does the cell generate the PMF?
Cellular
The end result, cellular respiration. So what are the 3 steps in cellular respiration?
Glycolysis, Krebs cycle, and
So if you were to describe glycolysis, how does that work?
glucose.
Break glucose in half. And what are the net results out of glycolysis? 2 pyruvate, 2 NADH, 2 ATP. You then go to the Krebs cycle. What happens in the Krebs cycle? Respire that pyruvate. And so per pyruvate, what are the net results from the Krebs cycle? 3 carbon dioxide, 1 ATP, 4 NADH, 1 FADH. You then go over to the electron transport chain. What happens in the electron transport chain?
Electron carriers.
Take the electrons to the electron carriers and pass them through the transport chain, causing protons to move across into the periplasm. And so protons in the periplasm is the PMF. Now we know that the PMF can be used directly by the cell for energy. So describe a process that uses the PMF directly. Flagella, which has that motor protein, the MOT protein, which uses the PMF directly for energy. But the PMF can also be used to make ATP. So how does, what would you need if the PMF is going to make ATP? The enzyme ATP synthase. Now, the electrons, once they get to the end of the chain, need to go somewhere. And where do they go? Onto the TEA. What is the best TEA that a cell can use? And that means they get the most energy out of it. But there are plenty of bacteria that survive on less than the maximum amount of energy. So what's the worst TEA that some cells can use?
CO2.
Carbon dioxide. What happens if no TEA is available at all?
Fermentation.
They can still survive on fermentation, getting those 2 ATPs out of glycolysis. Good. What else does a cell need?
The IV.
Inclusion body. And what is that used for? Storage. What else would they have?
The
Ribosomes. What are ribosomes used for?
proteins.
Makes proteins. And that's true in all cell types, but there is a difference between bacterial and prokaryotic ribosomes. And what is that difference?
Smaller.
Bacterial tend to be smaller, and why is that important?
Growth.
Makes it a good target for antibiotics. What else? The 2-component system. What is that used for?
Sense outside stimuli.
Sense outside stimuli. And how does it work? What happens if the sensor senses its stimuli? It leads to a phosphate cascade. What else?
Glycocalyx.
Glycocalyx. What is that used for?
Protection.
Protective layer around the cell. What are the 2 types?
Capsule and slime.
Slime.
And we talked about a specific example of one. What is that example and what does it do? Dextran leads to tooth decay. Good. We missing anything? Villi. They're a similar structure. Fimbriae. And what are villi or fimbriae used for? Adhesion. Now sometimes they're just used for adhesion, but sometimes they can be hollow in the middle, where they can then be used to transfer things from one cell to another. For example, there's a process of lateral gene transfer that requires a pilus. And what is that process? Conjugation. What are some other mechanisms of lateral gene transfer? Transduction, how does that work?
Oh, transduction is a virus.
Transduction is a virus. Transformation, how does that work? Reading. And then lastly, transposons, how do those work?
Jumping.
They're jumping genes. And what is the benefit of lateral gene transfer to bacteria?
Evolution.
Leads to genetic diversity, which over time leads to evolution. And genetic diversity is important for cells because it allows them to adapt to change. What's another mechanism of genetic diversity found in bacteria? Mutations. Good. The other thing that we need to build a pilus, specifically the conjugation pilus, then would be that plasma. We missing anything in this cell? That cell looks pretty complete, but now that cell is not just going to sit there and have all these parts. That cell is going to go through these active processes of growth and division. So it goes through a cell cycle. What are the 4 phases of my cell cycle?
G1, S, G2,
So how would you describe G1 phase?
growth.
It's growth, normal growth, or the growth period. And so it's making proteins, making energy needed for growth, such as mREB. What happens during S phase? DNA replication phase, so it makes its DNA. How does DNA replication work? You have the enzyme DNA polymerase, and what does it do? How does DNA replication work? So you take the DNA, split it apart, and you use one side as a template to build the other side, right? And that's possible because of base pair rules. What happens during G2 phase? Dividing metabolism, so it makes energy, proteins needed for division, such as FTSC. And then finally it actually divides. And so M phase is named for mitosis, the division process in eukaryotic cells. What's the most common process in bacteria? Binary fission. But there are others that exist, such as budding, exospores, fragmentation, stalk formation, and you should know how those work. As we allow these bacteria to grow and divide in a lab, we are often providing the optimal nutrients for growth, and therefore they go through a 4-phase growth curve. What are the 4 phases of my growth curve? Lag, log, stationary depth phase. So why do they go through a lag phase?
They're
Adjusting to their environment. Log phase, maximum growth. Stationary phase, maintenance, and death phase. Nutrients are now all gone. But that's in an artificial environment in a lab where we are providing the nutrients. In the real environment, bacteria have to find their own nutrients. And so we have different classifications based on how they get their nutrients. For example, there are 2 different ways to get carbon. What are those 2 different classes of bacteria with respect to carbon? Autotroph and heterotroph. So what does an autotroph use? Carbon dioxide. What does a heterotroph use? Eats its carbon. There are 2 different ways to get energy in the environment. What are those 2 different ways? Phototroph, which uses light. Chemotroph, which uses chemicals. And under the chemical subheading, there are the 2 subdivisions, organo and litho. And the chemolithotrophs are unique to bacteria. We then have 5 different classes of organisms with respect to oxygen. What are those 5 different classes? Aerobes, microaerophiles, facultative, aerotolerance, anaerobes. What does the term aerobe mean? Environmental levels. What would facultative mean?
It can grow without.
Can grow without, grows better with. What would anaerobic mean? Cannot survive in the presence of oxygen. There are 4 different classes with respect to temperature. What are those 4 classes? Psychro, meso, thermo, hyperthermal. What is the rough temperature range for mesophiles? Like 20 to 45. How about hyperthermophiles? Greater than 65. So if you were to categorize human beings under their carbon, what kind of carbon organism are we? Carbon organism.
Heterotroph.
Heterotroph.
No, heterotroph.
What kind of energy are we?
chemo.
Chemotroph. What kind of oxygen requirement are we? Aerobe. And what kind of temperature organism are we? Mesophile. Now that describes normal growth for a cell, and so we call that vegetative growth. But there's also these kind of specialized forms of growth. For example, when a bacteria lands on a hard surface, what happens to it? It forms a biofilm. And what are the hallmarks of a biofilm? EPS. If a cell finds itself under nutrient starvation conditions, what happens to it? Forms an endospore. And what are the hallmarks of an endospore? Exosporing on the outer core, and then SASPs and DPA in the inner core. What is the purpose of an endospore?
Dormancy.
It's a dormant resistant cell. What is the purpose of a biofilm? It's a protected community of cells. Finally, we can then look at ways to control bacterial growth, and there we're talking about either changing the physical environment or the chemical environment. With the physical environment, we primarily use heat as a means to do that, and so there are 2 different processes that are commonly used: pasteurization for food sources, or sterilization typically for medical equipment. How do those work, and what's an important difference between them?
Sterilization uses heat under pressure.
So sterilization includes pressure, and therefore sterilization kills spores. Pasteurization doesn't always kill spores. Finally, if we're talking about the chemical environment, especially if we're talking about the body, we're talking about antibiotics. And so if you were designing an antibiotic, what would you want?
Targets
a common pathway not found in human cells so that you have a broad spectrum antibiotic. So a cell wall, for example, is a very good target for an antibiotic. So give an example.